It is also a matter of pride that Dr. Anuji is the first lady resource person for this series. I feel extremely happy that we were students of the same degree class at CMS College, Kottayam during the late 90s. One could feel the brilliance and exploring capabilities of Dr. Anuji even in those budding years. It is no wonder she could move to alien lands and unknown cultures in pursuit of the best and achieve them. Only with awe I can see her marvelous achievements. Now, Dr. Anuji is a senior researcher in the Materials Science and Engineering Division in Product Development at Bristol Myers Squibb, New Jersey, USA. She has a PhD from ETH Zurich, Switzerland. She was a postdoctoral fellow at EPFL, Switzerland, and Durham University, UK. She is the subject matter expert for NMR spectroscopy at BMS, expanding applications of NMR from small molecules to complex and biomolecules. She is also a material selection and drug product development team lead for 6 BMS assets. She has authored 30 research publications, and she is the author of a kid's book titled My Story, a little boy's narration of his mom's journey across the world and as a half marathoner. Her achievements are beyond anybody's imagination, and people like Energy offer our generation the necessary challenges to excel. With a wholehearted joy, I welcome today's resource person, Dr. Anuji Abraham, and she'll be giving us a presentation on the topic, the role of enema in the pharmaceutical industry and opportunities for chemists. On behalf of everyone present here, I welcome Dr. Anuji to lead the webinar. Thank you.
Thank you so much. This was beyond my expectation of introduction. That has been so nice. Yeah. It just my mind because I was remembering all those 1990s, right? Anyway, this is too much. So let's get started. So much.
Okay.
Thank you for inviting me over here to give this talk. And it's a pleasure to be here. And as I was telling you before, that I was really excited because this is really coming back home and sharing the experience with you guys. I started in Kottayam in CMS College, so this is a great opportunity for me to share my experience beyond the technical aspect, just to speak about what we can do, starting in Kottayam. So that's where I'm really looking forward. Yeah, let's get started. So as Sheetal was telling, I am part of material science. I'm a senior researcher in material science and engineering in the product development at Bristol Myers Squibb. So can you all see my slides well?
Yeah.
You can see my slides well? Yeah. Okay.
Yes. Yeah.
Okay. So let me keep an outline of what the presentation is going to be. So first of all, I'll take a couple of the slides actually to give a flavor of the career journey I had and how I reached and Bristol Myers Squibb. So then I would like to give you a flavor of pharmaceutical discovery and development, a little bit about the drug substance, the formulation, drug products, role of material science and engineering in pharmaceutical industry. And coming to the last is the opportunities NMR spectroscopy has in a big pharma, with some examples. But this is all, not going deep into things, but just to give a flavor of what we can do, right? So before going to the main talk, I would like to say that, this is where I started. As I was telling, I studied with Sheetal and many other great, friends of mine at at CMS College, did BSc. Then I moved to do Masters in Madras Christian College in Chennai. After that, I wanted to do PhD abroad rather than doing a PhD in India, which is also great. But I decided to pursue a PhD abroad. I started applying in Europe, and I got accepted to do a PhD at ETH Zurich in Switzerland, which I spent about less than 4 years doing my PhD there. Then I decided to move further, doing few postdocs, in fact, and starting with a postdoctoral research study at EPFL Lausanne, and then to Durham University in the UK and as in WVU USA. Before joining BMS, which is about 9+ years ago. So it has been, it has been a journey, right? The reason I wanted to highlight this was like, it's not just PhD, the postdoc, all of those experiences giving you that world experience, right? It's not just a job, it's a career. So you develop your network and eventually, I am now at BMS, which is, great. I mean, it's a great place to work. So just a little bit more of who I am. I didn't realize that Sheetal would give an elaborate introduction of myself, but just to give, I'm a researcher and a subject matter expert of NMR spectroscopy at BMS. I'm a project leader and I'm also a people manager. I have people reporting to me, but I'm, just beside the internal, involvement at BMS, that's my main job. I am also involved externally. Can it be like, Panic NMR conference? I'm in the executive board of directors. This is just to give a flavor of what we can do, because it was important for me to show this here, because I hope some of you perhaps might think of taking this as your career path and want to give a flavor of what you can, right, in like 15 years of time. And so I co-advise a PhD student at University of Liverpool. I'm a reviewer for many journals. I have some publications. And first, and just want to highlight one of the most passionate thing I do and I continue to do is mentoring. Actually, I mentor junior scientists within BMS and externally. So I have students from Rutgers, around New Jersey. We do mentor, and I am a mentor as well. And this is something which I really am passionate about, especially female mentoring, because that's all over the world. It's not just India, right? All over the world, that's always like not equal, right? It's disproportionate, and I want to share, like many of my other colleagues, I want to share my experience and give that motivation to female students as well. No, not excluding males, including males. So here is my LinkedIn profile. So if you wish to, connect with me, please add me through LinkedIn, or here is my email address. I'm just giving you right there. So if somebody would want to reach out to me, please directly reach out to me. I am happy to help you in any sort of career advice or questions you have. So that's about me. And with that, I'm going to go to the main part of the talk, which is, as I was telling, Bristol Myers Squibb is a big pharma. And meaning that this is the whole process of pharmaceutical development, right? So it's very high level. You start with, in discovery, identifying thousands of molecules to find very few hundred hits. And out of that 100 hits, only 1 to 3 will be seen as developable and would go into development. That's what I am seeing. I sit in the product development, meaning that it's further moved from the discovery phase, right? So when I get, it's just few molecules which I get, which has to go into a product and go into clinical studies as shown here. You have to do preclinical studies in animals, different animal models, show the toxic effects or whatever, and then you have to take it to in clinical, different phases of clinic, which is phase 1 and 2 and 3. For instance, just to give an example, our COVID vaccine now is in phase 3 development, right? So phase 3 clinical study, and which has been approved in the UK, the Pfizer vaccine, and we are waiting for the approval in the USA, right? Hopefully it will be approved in India and other countries too. So this is in the phase 3 phase, meaning that as you move through the phases, the study is getting bigger and bigger. You saw the number of patients which were trialed in the phase 3 studies for the COVID vaccine, right? So the study gets bigger, and eventually, you show the safety and efficacy of this through these clinical studies to get it commercialized, to get it approved and commercialized. Even then, it's not starting. The research is never starting, right? We are R&D, meaning that there is life cycle management. You can't just abandon a drug just by commercializing it. You always have to have, look for problems or anything which could come later on as well, right? So there's always an eye, which is called the life cycle management. So it's a huge process. Typically, it takes like several years for a drug to get approved, meaning that it's from 5 to 10 years. It's shrinking actually, but for instance, this COVID is one of the examples. We have taken a drug, we have developed a drug, right, a vaccine, and got it approved within a year, which is like very fast track, which is very, very unusual. This is this is a very emergency condition, right, where the approvals and the studies went super fast. But typically it takes like 5 to 10 years for a drug to be approved. So with that, I just want to show that this process of drug development is a very highly collaborative effort, meaning scientists from discovery to development to commercialization, manufacturing, clinicians, all these people they are matrix teams. So when I said that I was leading a team, it's a group of people I lead where we discuss and how to bring this drug substance as a drug product to commercialize it, right, through the whole journey of it. And so collaboration is key in this process, right? There is no, transparency, collaboration, open communication is very, very important during this process. So coming to the types of drug substance. So drug substance can be a small molecule, small organic molecule, right? It can be complex molecular assembly as shown in the middle figure, or it can be a very large molecule, like a biomolecule, proteins, which we call biologics actually. So there are different, the size can increase from a small organic molecule to large protein with 150 kilodaltons in molecular weight. But this drug substance, what we have as drug product is just not the substance. You have to formulate. The process is called formulation, meaning that the drug substance, which is the active ingredient, has to be mixed with excipients, which we sometimes call fillers, in the right ratio to give the best stability and bioavailability to the drug product. So excipient is, we call it fillers, but they are not really fillers. Every excipient has a role. There are, some of them are used for disintegrating, some of them are used for, everything has a role. There are functional excipients or non-functional excipients, but they do have a role. It's essential for ensuring that the active ingredient of the drug is delivered to the correct part of the body in the right concentration and right rate. So that's formulation. That's very high level. What's a formulation? So that brings us to the drug product. So BMS is focused mainly on orals and injectables. And there are a lot of other drug products such as the topical, transdermal, or inhalables. All of that is there, but we focus mainly on the orals and injectables. Orals meaning that it could be a tablet or a capsule, solution, suspensions. In pediatrics, it's mostly like suspension-based products, right? And then it can be an injectable, like intravenous. It can be IVs, or subcutaneous, like quick injections, or intertumoral, like we work on oncology, so it can be intertumoral as well, so directly injecting into the tumors. And when it comes to the product can come from any sort of drug substance. For instance, the orals can be a small molecule oral product, right? It can start from a small molecule, or it can be the complex molecule, which is making the oral product. But if it's an injectable, it can be a small molecule or a complex molecule or a biomolecule. Anything can come as an injectable product. And for the other side, it's mostly like small molecules and complex molecules, which is going into the different routes of administration. So what are the key attributes? So there are three key attributes to the whole process of drug development. Number one is safety. Safety has to be there for any product or the substance which goes into the product which is being developed has to be safe. And that's why the clinical studies are coming in, right? The safety. The second is the efficacy. It has to be shown that it is efficacious for the kind of disease we are trying to cure, the kind of indication, right? So it is important to show that drug is safe, it is efficacious, and the third, but very importantly, it has to be shown that it is physically and chemically stable, because the drug has a certain shelf life. It is typically from 1.5 to 2 years, and it is important for us to show that the drug is stable chemically and physically during the shelf life of the drug. So that's where the research is coming, right, to understand those three key attributes. So where is material science and engineering sitting? I'm from material science and engineering division, the product development. What do we do? So our core thing is really understanding the physiochemical characteristics of the drug, characterization of the drug substance and the drug product. We give understanding into the structure and the dynamics of the molecule, the nature and the stability of the substance and the excipients. Sometimes, as I was telling, the excipients can be functional excipients. We do understand the excipients. We give understanding into drug excipient interactions. So I'll give some examples as before, right? I have examples to show some of these aspects. So what kind of tools do we have, right? This is coming back to our chemistry and physical chemistry, right? I would call myself a physical chemist or a material scientist. So this is where we can connect, you guys know, right? There is the physiochemical characterization tools could be diffraction methods like single crystal X-ray diffraction, powder X-ray diffraction, can be spectroscopic methods like nuclear magnetic NMR reference, NMR spectroscopy, or vibrational spectroscopy like IR and Raman infrared and Raman spectroscopy. Mass spectrometry is there. There is imaging methods. There is NMR imaging. There is Raman imaging. There are thermal methods. There is like calorimetric methods, gravimetric methods, the microscopic methods. There are particle size methods like laser light scattering, optical methods, and there are others. I just put there's a whole lot of other things we can do. Oh. There is a message coming for me. Just let me just see. Oh, okay, sure.
Thank you, ma'am.
Yeah, no problem. Is it better now?
Yeah, better now.
Okay.
Thank you.
All right.
Thank you.
No problem. So all of these methods are there. And besides that, we also do molecular modeling. So because some of the times it's not just experimentally understanding, we can understand these molecules and the dynamics computationally as well. So it goes hand in hand. So everything is part of material science and engineering. So what are the opportunities for NMR spectroscopy in big pharma? So this comes to what we have in the centers of excellence of NMR and BMS. So we have NMR facility all across the world, multiple facilities in New Jersey. We have one in Morton and one in BBRC, India. So I just wanted to put it out there because we have a site in in Bangalore. And besides that, we collaborate with external academic partners. For instance, we have collaboration with Princeton University, University of Delaware, and Bruker Bioscreen to do some high-end experiments. Because some of the times we might not have to, these machines are like millions to $10 million, budget, right? So some of the times we might not need to have the machine in-house. So we collaborate with these places to have access to these facilities. And that's what we are. So where I am is actually, I oversee all the solid state NMR activities all across the world for BMS. So coming to the basic principles of NMR. First and foremost, for NMR active nuclei, the spin quantum number has to be greater than 0. I'm not going to explain too much into what a spin quantum, I believe all of that is the basics, and we are chemists, physical chemists, we know that, right? So if you look at across the periodic table, we can do NMR on pretty much all the nuclei, right? You can see that there are spin halves and spins greater than half, which are all colored here. So there is, for every nucleus, there is at least one or more isotopes which are NMR active. So that's good news, right? So we can do carbon NMR, nitrogen NMR, oxygen, fluorine, protons, correlation NMRs, all of that, right? That is great. So again, coming to the very basic principle, what it does is we have a spin, for instance, let's take a carbon or a proton, which has a spin of half. So NMR is working in the presence of magnetic field, right? So without the magnetic field, the energy levels, the spin energy levels are degenerate. In the presence of magnetic field, it loses the degeneracy, meaning that is called the Zeeman interaction, which is happening, which you can see here. And under the influence of radio frequency radiation, it can excite the spins. And when the spins relax, it's called a free induction decay, which is Fourier transformed, and you have the NMR signal, or the peak, or what we call as the NMR spectrum. So likewise, if you have 3 different atoms in the molecule, each of those spins will excite, and it will give rise to 3 different peaks in the NMR spectrum. So that is a fingerprint of a molecule, right? So it's basically giving a fingerprint to the molecule. So this is showing how a spectrometer would look like. I just wanted to have like a feel of it. So I don't know how many of you have seen an NMR spectrometer. This is how the NMR spectrometer looks like. There's a magnet and there's a console which is behind it. So this is the magnet. This is a schematic representation of the magnet where the probe is going in and the sample resides inside the probe here, right here. And you can have different sort of sample holders depending on the kind of sample we are dealing with. Can it be solids or liquids or anything in between, right? Gels, etc. And these are the different sample tubes. This is just to get a feel of what it looks like physically. But why NMR? So as I was telling, it gives the fingerprint of the molecule. Can provide quantitative, structural, and conformational information into a wide range of compounds. It doesn't matter what size the molecule is. It can be small molecule, complex molecule, biomolecules, excipients, etc. It can be done in solid and solution state. It's very sensitive to the local environment. I'll give you some examples where we can see that. The environment and the dynamics, NMR is very sensitive. It's a primary tool for characterization of materials agnostic of molecular modality. So NMR in pharma. So this is showing how NMR can be used across pharmaceutical company. Can it be discovery or development, right? And some of these problems are very much coming from development, but it can be used for structural determination of API, excipients, degradants, metabolites, as I was telling. A lot of things can reside in discovery or development. It can be used for metabolomic profiling, chemical purity identification, phase purity identification, physical phase purity identification, polymorphism, chemical composition of small molecules, the sample heterogeneity, meaning that if a sample is in crystalline or amorphous state, NMR can give quantitative information about that. It can predict stability in amorphous dispersions. I'll give some examples for you to understand what I meant by that. Probing API polymer interactions in amorphous dispersions. Spatial distribution of multi-component complex systems. Just again to give you a close example is lipid nanoparticles. Our COVID vaccine, again coming back to that, it's a lipid nanoparticle which is filled with miRNA strands. So it gives the whole arrangement of, it gives the spatial distribution of these multi-component system in these lipid nanoparticles. We have very similar system at BMS. Unfortunately, I cannot give you like all the things we are doing at BMS because of proprietary reasons, but I have shown some which I could talk publicly, right, which has been accepted and gone through the lawyers. We can determine higher order structures for large molecules, biomolecules like monoclonal antibodies, proteins, antibody drug conjugates, etc. It is also used for preclinical imaging. As I was studying, there is NMR spectroscopy and NMR imaging. So it's very, very heavily used for preclinical imaging and tablet imaging as well. So coming to the first example, it's about polymorphism in small molecules. I hope I was there live to ask how many of you know about polymorphism, but I can give you a brief overview of what it is. So a molecule can exist in multiple crystal structure, right? The same molecular structure can have different conformations in crystal form. That is called a polymorph. So this is a schematic in a pictorial representation of this. If you consider this as one API molecule, it can sit and pack in different arrangements, which leads to polymorphs. But polymorphs, it's not just API by itself. It can also sit together with a water molecule or a solvent molecule, which can also have polymorphism, meaning that depending on how they are sitting together in different arrangements leads to polymorphism in a solvent or a hydrate. It can make complexes, meaning that the API molecule can complex with non-active ingredient and make complexes, and they can arrange in such specific ways, in different ways, different packings. Same, it can make co-crystals. It can have a co-former, can sit together with a co-former and have different arrangements. So this is why is it important, right? So yes, API can sit in different conformations. In oral product, you have crystal form. Most of the time, we have crystalline material, right? But why is polymorphism important? It is important because, as I was telling, different crystal structures have different properties. So chemical stability can vary depending on the crystal form. The solubility and the bioavailability can vary. All of that is impacting that three key attributes, right? You are thinking about the toxicities of, let's keep that to the side now, but bioavailability and the stability aspect, right? So this is really, impacting the chemical stability, the solubility, the bioavailability, hygroscopicity sometimes, meaning that it can impact the storage conditions, again, going back to stability. Morphology, it can impact the processability, meaning imagine like some of these forms can be just needle-like forms and some others can be like spherical crystal material, while some others can be needle-like. So it is heavily, it is very difficult to handle needle-like material in comparison to spherical material. So it impacts the processability of the material. There are ways to tune them, but this is like very high level showing how the direct impact of the change in crystal form onto different aspects, right? And the powder properties again can have impact on the processability. So how is NMR being useful here? This is again coming back to our link to the NMR. So NMR gives the fingerprint of a molecule, right? So if for instance here, there's a simple molecule which is gabapentin, and it has several number of carbon atoms, and this API can exist in different forms, which is called as, let's call it form 1 and form 3. And depending on the conformation of these, how this molecule is in the crystal structure, the pattern, the NMR spectrum varies. You will have the exact number of carbon atoms, but the environment of those will be different depending on how the conformation is, and that gives a different fingerprint for each of those polymorphs. And you can also look into mixtures of polymorphs, right? So that's one of the key things in small molecule work. What we do using NMR and single crystal X-ray diffraction is really understanding the polymorphs and phase purity. So going back again to the polymorphism, sometimes we want to know how many molecules are in the asymmetric unit of the crystal structure, meaning that how many molecules are the repeating unit, right? So this is an example of barbital. This is also taken from a textbook. These are very close examples of what we would do at EMS, but this is taken from my previous advisor. Actually, it's all in the book, but just wanted to show you how these are exactly what we would do in our company, right? So here it's showing that, for instance, this barbital can arrange as repeating one molecule or two molecules together. So meaning that NMR spectrum varies depending on if there are one molecule versus two molecules, meaning two molecules you will have multiplet of, or doublet of all of the peaks, which shows that there are two molecules in the asymmetric unit, and barbital 3 has one molecule in the asymmetric unit. So that's another use of NMR in the small molecule world. Coming to a second example, which is a little bit more complex, it's understanding phases and phase separation in liquid crystal drug delivery system using deuterium solid state NMR. This is a published work, so I can talk about it fully. So, little bit of background on these liquid crystals. They are self-assembled systems, three-dimensional network, liquid crystal network, which is formed by liquids like SPCs and GDOs. When in contact with water, I'll give details later, it makes this beautiful three-dimensional liquid crystal networks. It can sit as lamellar, layered, or hexagonal phases, or cubic. There are different ways it can arrange. And the key thing is the drug molecules, the active drug molecules, can it be small molecule or a little bit more bigger molecule, which we call as milli-molecule, can be incorporated into the structure or between the layers in the hydrophilic layers, depending on the property of the API. If whether the API is more hydrophilic or hydrophobic, it can be embedded into these layers, and which has excellent in vivo encapsulation and controlled release properties. It's highly useful for drug delivery systems for controlled release subcutaneous injection. But the challenge here is the characterization of the system, because it's highly complex. Number one, it's highly mobile, it's amorphous in nature. So techniques like X-ray crystallography cannot really help. That's where NMR spectroscopy can play a role. So here, just going a little bit into the in situ gelling principle. So you have this SPC, GDO, which are lipids, phospholipids, which is mixed in the solvent and the drug, and water penetrates. It makes these gels, and eventually the drug is released out of this gel, which is made in situ, which is in the, you are injecting it into your skin. It's subcutaneous, and the drug releases with time. So the desire is to have a controlled release for the drug inside your body. So preparation. So for our study, this is to understand how the release is happening, what are the phases which it's making, right? So what we did is we had this phospholipids, SPC and GDO, which the structures are right here, mixed with ethanol, but instead of using water, we use D2O. It's deuterated water. So we are leveraging deuterium NMR to understand this, right? So it's again making these different layers. So if you look at a little bit more on the deuterium, think about it as a fingerprinting, right? So deuterium is a protopole nucleus. I'm not going to go into the details. It has spin 1, meaning it's not a simple nucleus to deal with. It is a little bit more complicated than like a proton, which has spin half, or a carbon, which has spin half, or a fluorine, which has spin half. This is a little bit more complicated. So there are 3 things which are defining so instead of having a beautiful, nice line, what you would see for deuterium is like these patterns, or these different patterns. So these patterns are defined by 3 different things, right? Think about it like that. So one is the protopole coupling constant, the asymmetry parameter, and the electric field gradient. Out of those 3, 2 of them are influenced by the environment by itself. So meaning that if the environment changes, the pattern can change because it's influencing the asymmetric parameter, or the electric field gradient, which leads to a change in the pattern. That's kind of fingerprinting, actually, right? So this is how we probe it. So we have, this is the phase diagram for SPC, GDO, and water system. And you can see these blocks here, which is showing the different phases. For instance, you have the micellar phase here, the cubic phase, the hexagonal and the reverse hexagonal phases here, the laminar phase here. And depending on where the phase is, you have a different line shape for NMR. So that's what we are probing. So we probe through the, we have probed a lot more, but this is just to show a nutshell of what we probe. And you can see the mixtures, where the transitions are happening. And like this, we could help to choose the desired, phase in order to have the right release for the drug. So that is in a nutshell what NMR could provide here. So coming to example number 3, I'm just looking at the time, is probing API excipient interaction. This is also a published paper in Molecular Pharmaceutics. So, if you are interested, you can have a look at it. It's, so why API polymer? Why polymer here, right? So consider polymer as an excipient. So what is happening here is, most of the time in oral drug products, the drug substance, API, is in crystalline form. But sometimes crystalline form is not the most desired form because of many reasons. So one is the bioavailability by itself. Sometimes it could have food effects or pH effects. There are a lot of other factors which comes into there. So we need to develop an amorphous system. But amorphous systems are always metastable systems. It can easily crystallize back. But we want to keep the amorphous as amorphous. How we would do is disperse this amorphous API in a polymer matrix. So that's what is depicted here. You have these matrix of polymer and you want this API to be dispersed uniformly in this matrix in order to have the right property eventually, right? You need to have the single phase because you don't want phase separation and eventually API crystallizing out. You want that uniform, nice system, which is the yellow bit here, right? That's what is desired. How do we probe that? Because again, here we are dealing with an amorphous system. So X-ray crystallography is down the list or not in the list of understanding such a system, right? So that's where spectroscopy is coming in and NMR spectroscopy is coming in. Can we probe that this is a single phase? Can we see API polymer interactions? Yes, we can. So here, this is an example I'm showing here is an API which has fluorine in it. So you can see that there are 3 fluorines, CH3. And because this is only with the API, the fluorine atoms are only with the API, the polymer is HPMCAS, which does not have the fluorine atoms. So you do a carbon-fluorine correlation NMR and try to understand which are the correlating peaks, proximity-wise, and can say that there is API polymer interaction happening in the system and how uniform it is, right? All of that can be obtained from doing this kind of experiments. Coming to case study 4, the system is getting bigger and bigger now. In this particular case, NMR was used to provide understanding into higher order structures of monoclonal antibodies or MAPs. So why understanding this sort of system, right? They're very complex. They're complex 3D structures. So one of the key things we have to think is because it's basically amino acids, right, and peptides, peptide links, and they can fold up to infinite number of ways into elaborate shape to have this vital function. So depending on how the conformation is, the function of this map can change. So the rearrangement of these molecule, understanding the rearrangement is very, very important because it could have like very catastrophic effect on its function. Sometimes it can be not functional at all depending on the conformation. So it's important to understand the regions of unfolding, the regions of aggregation, the chemical degradation. All of that is very important to understand its function and its stability. So that's where NMR is coming again. This is a work which I can talk about in one presentation by itself, but I'm just giving you in a nutshell of what we can do. Again, as I was telling, NMR is a fingerprinting technique, right? So you can do carbon NMR and proton NMR. Here we have done carbon-proton correlation NMR to understand the map, and then you stress the map in different conditions and see the changes and assign the regions where the changes are happening in regards to conformational changes or in regards to real chemical degradation or chemical changes. And it's really probing the regions and giving information into the function and stability. So this is one slide I'm showing. The right-hand side is showing some of the modeling work we have done and how the NMR information is correlating with some of the scores we have from modeling. And we have done some BC analysis. This is much more elaborate, but I don't want to go into the details, but want to give a flavor of what NMR can do in this big, large systems as well. Again, I want to connect you with something which was in the news a couple of days back. Hopefully you guys have seen it, I guess. This was about predicting how a protein folds into unique 3D shapes by DeepMind's AI. So this is a mystery which has been solved because, as I was telling, it's heavily important to understand the folding, right? So if you can do it computationally, it's great, right? Because doing it using NMR or X-ray crystallography is challenging. It's the previous slide showed it as very simple, but it is challenging. It takes a lot of designing of the experiment and assignment, and all of that is complicated for such large systems. So if you have read the article, they did mention about NMR and X-ray crystallography being important tools for understanding protein 3D structures, right? And yeah, so what is the next there, right, for AI or the NMR? Just wanted to correlate to what in real life, I mean, we are doing real life, but it's much closer when something comes in the news, right? So understanding how a spike protein on the surface of the SARS-CoV-2 virus interacts with the receptors in human cell, that's something where the AI, the computationally can be done, or NMR or crystallography can be done through that as well. So just wanted to give that as well out there. And with that, I am concluding my talk, and which brings me to the acknowledgements. I would like to acknowledge BMS. I mean, I don't want to mention names. It's the whole company I want to acknowledge for the opportunity, the great place I am working, all the collaborations, all the freedom of work I have beyond the industrial research, how we can connect externally and do research. It's a great place to work. And I just want to mention that when I was moving from academics to industry, I was talking to one of my PhD advisors and asking, is that a great thing? Because in my mind, half of my mind was always like moving into academics, right? And my professor was telling, if the company gives you opportunity to research and everything, go for it. And I did go for it, and I do not regret now, right? Because it's been great. I have all the opportunities how an academician would have, through an industry, because perhaps it's big pharma. But I want to acknowledge BMS, I want to acknowledge ETH, EPFL, Durham, and WVU for all the opportunities I've had for the NMR career. And not to mention, I want to, mention MCC and CMS College, but, this is where I started my NMR career, and I want to acknowledge these institutions. And some of the pictures I've taken from different parts of the world, actually. That's ETH on the top there, and there's BMS on the right there. So with that, I'm concluding. I'm happy to take any questions. And here again, there's my LinkedIn. So feel free to add me, and I am always happy to give any sort of, advice, career questions, anything you may have.
Thank you, ma'am.
Oh, not at all. Pleasure. Yeah.
It's time for questions.
Sorry.
It's time for questions now.
Yeah. Yeah. Yes.
Hello,
Yeah.
ma'am. Can you hear me?
Yeah, I can hear you. Can you hear me?
Okay. Okay. Yeah, I can hear you, ma'am. Participants, it's time for questions now. You can unmute your microphone and can ask questions to Anuji. Please.
No questions? I don't think there are any questions. But feel free to ask me questions. If you think of something later, you can email me and ask. If you are not want to ask now, please feel free to email me and ask. Technical questions, anything.
Hi, ma'am. I'm Jinda. I had emailed you yesterday. Can you hear me?
Yes. Yes.
Okay. It was indeed a great pleasure to have you here with us today. And I would like to say, above all, you will remain as an inspiration to all of us. I just want to ask, because of curiosity, some doubts about the drug delivery systems. Have you ever tried with any inorganic hybrid materials? What are the
Yeah.
possibilities, or what are the features in means reaching how whether we can use such systems for drug delivery?
Yeah.
Because I had been working for my
Actually, yeah.
uh,
Yeah. Go on, please.
for my PhD. I had been working on porous materials, so transition metals combined with nucleobases, and also I just wanted to know how can we reach the goal, like whether I can use it or any possibilities remaining.
Yeah.
And I have found some systems they have tried. So
Yeah, so definitely, right? So that's, thank you for that question. Yes, it is relevant. So here I showed that it's a phospholipid system where you make this liquid crystals, right? But as you pointed out, you can have inorganic materials. So we do have something called we don't call it liquid crystal. That doesn't exist as liquid crystal. They are co-processed materials, right? With inorganic
yeah,
materials like silica and silicon-based inorganic materials. And we also have
okay.
gold-based nanoparticles, right? So for instance,
Mm-hmm.
you can have like gold nanoparticles, you have amino acids or peptides around it, which links to your drug, right? So there are
Yeah, yeah, yeah.
several. So this drug delivery by itself is like a huge area we are working on. And I just
Mm-hmm.
give you one example, right? There are several things you can do. Going out,
Ma'am, it's not audible here. My team is good. Could you please mute your mic? Okay, thank you. Ma'am, please.
did you hear what I
No,
said?
the last sentence I didn't. Sorry.
Yeah, so I was telling that as you pointed out,
Mm-hmm.
the drug delivery system, drug delivery itself is a huge area we work on. And I just
Okay.
give you one example of this liquid crystal system, right? We have liquid
Okay.
nanoparticles, co-processed materials, as you mentioned, right?
Yeah, yeah.
That's the thing we work on. So there are several ways you can get that, right? But yeah, was happy to talk. I actually have a publication on gold nanoparticles as well. But I'm happy to talk. We can take it offline if you have questions, anything, something you are
Mm-hmm.
researching on or whatever it is, I'm happy to talk about it. Yeah.
Okay, okay. Thank you. Maybe I will contact you later.
Absolutely,
Thank
yeah.
you so much. Thank you so much.
One question, ma'am. A little doubt. How far the modeling techniques helpful to the drug delivery system?
Sorry, I just got disconnected for a moment. I'm back. Was there a question?
Hello?
Hello, I'm here. I got disconnected for a moment. I'm back now. Sorry, apologize that. Was there a question?
How far the modeling techniques helpful to our drug delivery system?
Oh, good question. Yes, it is very helpful. For instance, I can give you one example, which I couldn't show you, but let me tell like a very high level, right? We have a lipid nanoparticle, which is very much like the COVID vaccine, right? So we have the lipid bilayer and the API is encapsulated in this bilayer, and we have the targeting agents, which is sticking out, right? So that's the agent which is bringing to the right location inside. It's an IV actually. So in that case, we wanted to understand using experimentally and computationally how the bilayer is, right? So modeling gave a lot of information into the bilayer information, which was supported by experimentally as well through NMR-like tools. And we also wanted to understand the location of the targeting agent. For instance, is the targeting agent, because it was, I can give you a very high level again, it was divitamin A, it was a vitamin A moiety, and we wanted to understand whether all of that is it partially inside, partially outside, how is it sitting on over the layer, is it like partially fully inside, all of that, right? We had a lot of information from molecular dynamics actually. We did molecular dynamics, got a lot of information which was supported experimentally as well. Yes, we can. But those are very intensive calculations, right? So it might take a lot of computational power for that, doing such things. Same for maths, right? Anything in biologics, it takes a lot of time. Small molecules are easier, but even for small molecules, right? Some depending on the system, it can, can be computationally intensive. So that's the only bottleneck. But yes, we can do it, and we do it. Yeah.
Thank you, ma'am. Thank you. Ma'am, is there any molecular docking studies in your lab?
Can you elaborate on that? When you said doping?
Protein specified molecular docking studies?
Oh, dock. No, we don't do docking studies. It's mostly coming in discovery. Yeah, so that's sort of, key mechanisms, all of the receptor stuff, the linking, all of that is done in discovery. Yes, we do it, but not in the development area, but it's all done in discovery. Yes, we do.
Thank you, ma'am. Thank you very much.
Any more questions? If anybody wants to ask in Malayalam, please be free to ask.
You could have had all Q&A in Malayalam. But as I was telling you before, right? Feel free to reach out to me, right? I'm more than happy. This is one of my passions actually, to connect and, share experience, give career tips, whatever, right? Can it be technical or not technical? Please feel free to reach out to my email. It was there, right? I can share it. Maybe Sheetal, you can share it with the people, the group here, if you're okay with it.
Yeah, of course, ma'am. We can share.
Participants kindly fill the feedback form which is shared in the chat box. This is for issuing e-certificates for the participants. I think questions are over. That was indeed a great session. I am sure that we have heard a lot of new arena which we are totally unaware of. Let me hold on record our immense gratitude to Anuji Abraham for her informative talk. Now, Dr. Priya Thambi is here to deliver the vote of thanks.
Good morning to all. As all good things come to an end in life, so is this webinar also. Respected Head of the Department, Dr. Subhambino Thomas, our Chief Guest of the Day, Dr. Anuji Abraham, all the participants gathered here, and my dear colleagues. Today's webinar was with full of knowledge and energy packed with interesting things. It gave deep insights into the topic and also revealed interesting facts regarding the steps involved during the drug release. And the point Dr. Anuji Abraham told us was about the applications of NMR in pharmaceutical industry, and it was really informative, ma'am. I'm pretty sure that the precious knowledge you have delivered gave us will definitely help us in our studies in future. Once again, I would like to thank Dr. Anuji for taking out time from her busy schedule and enlightening us with her knowledge. Thank you so much. Thank you for sharing those lovely flavors to our participants also. Next, I would like to express my sincere gratitude to our principal, Dr. Biju Thomas, for giving us permission to organize this webinar and for his enthusiastic support given to us. I would like to propose vote of thanks to our beloved Head of the Department, Dr. Subhambino Thomas, for her constant encouragement and support she has given to make this moment a great success. From the bottom of my heart, I express my sincere thanks to Dr. Jinta Thomas, coordinator of this webinar, for her hard work. Your conference has been a great success and in all aspects. A special thanks to the organizing committee, teaching and non-teaching staff of the department for their unflinching support for this seminar. Our heartfelt thanks to all the participants and students for their active participation. Finally, I thank the wonderful students who have turned up in such great numbers, not only from our department, but also from other departments of various colleges. Thank you so much for your cooperation. And once again, I thank all for your cordial cooperation. Thank you.
Thank you, Priya. Next and the last session of this webinar series will be on 11th December at 4 p.m. by Dr. Prashant Wilfred on the topic water splitting. Expecting the participation of all in the next session also. Hence, we come to the end of a very fruitful discourse. Thank you, one and all. Have a nice day. Thank you.
Thank you, everyone.
Thank you.
Thank you, Anji.
Talk to you later.
Thank you, ma'am. Thank you. Call you.